DNA replication starts in the certain part called origin. Usually this part is identified by certain DNA sequences. There can be multiple origin within a DNA. Its origin helicase, the unzipping enzymes comes in and unzips the DNA. SSB proteins that stands for single-stranded bindings, proteins behind to DNA strands and they keep them separated primers come in and makes RNA primers and both strands. Remember, that's really important because otherwise when DNA polymerase comes in, it would know where to start. Now, DNA polymerase can get to work. Remember, it's an important enzyme at DNA bases. Now, you have two strands, right? They are not identical, they complement each other. They're also anti-parallel, so that means they really go in the same directions. We don't say a strand goes north and south. The direction of DNA strands are little different. We say that DNA either goes by which prime or prime. This seems very confusing. What the word is that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons and the sugar are numbered right the oxygen in a clockwise direction. 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. 5 prime carbon is only outside the register. May do the same thing to other side, but keep in mind that strand is slipped just because the DNA strand is anti-parallel to each other. So let's count this again, clockwise. From the oxygen, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And the 5 prime is out of the ring. This strand on the left wants 5 prime to 3 prime, and the strand on the right here wants 3 prime to 5 prime. Well, it turns out that DNA polymerase can only work in the 5 prime to 3 prime direction. So, the strand runs 5 prime to 3 prime. It's fine it's called the leading strand, but the other strand make a little tricky. DNA polymerase can only go in the 5 prime to 3 prime directions. So RNA primers it has to settle have of extra primers due to do that shown here. It takes longer too. This strand is called the lodging strand, which is pretty fitting. On the lodging strand, you tend to get these photo fragments of synthesized DNA. They're called Kazaki fragments. Amazing name. The primers have to get replaced with DNA bases since the primer is made of RNA ligase. Remember that gluing enzymes has to take care of the gaps of Okazaki fragments. And now, in the end, you have two identical double helix DNA molecules. From the one original double helix DNA molecule, this semi-conservative replication because the two copies each contain one old original strand and the newly made one. Oh, one last thing. Surely you have to proofread your works before to catch errors. Well, you definitely don't want DNA polymerase to make an error. If it makes the wrong DNA base, then you have incorrectly decoded gene, which could ultimately end up in an incorrect protein or no protein. DNA polymerase is just awesome. It has proofreading ability, which means it rarely makes mistakes, and that's a good thing.